Welcome to our webinar on using double helix optics light engineering for three-dimensional single particle tracking. In our last webinar, we gave you an overview of our technology and product offerings. In this webinar, we'll dive deeper into the different applications of single particle tracking using our engineered PSFs. Single particle tracking has found applications in various sciences. For example, in life sciences, it has been used in cell signaling and studying transport mechanisms for mRNA. More recently, single particle tracking has been used for targeted drug delivery and for gene targeting in applications like CRISPR-Cas9. Single particle tracking in microfluidic channels is also gaining momentum due to the greater control afforded over the assays, reducing reagent requirements and the corresponding increase in reaction times. In fundamental sciences, single particle tracking has been used for studying interactions and diffusion characteristics of molecules near surfaces. Other applications include verifying stability of molecular traps like optical traps or Abel traps, which are typically used to quench the Brownian motion to allow for prolonged observation of these molecules. Furthermore, by labeling the surfaces of polymer structures, their shape and size can also be studied using single particle tracking. Last but not the least, tracking of fiducials on a cover slip for active and passive drift correction is essentially single particle tracking in itself. In this webinar, we'll show how, unlike traditional approaches, single particle tracking with our technology can provide axial super resolution with an extended depth of field, and all this without the need for axial scanning. Commonly known competitive techniques to single particle tracking include fluorescence recovery after photobleaching, also known as FRAP, and fluorescence correlation spectroscopy, also known as FCS. However, both these techniques only provide average aggregate information, and function-dependent heterogeneity is lost. And although they provide millisecond temporal resolution, that spatial resolution is still diffraction limited. Now let's look at what a typical single particle tracking experiment entails. The first step, of course, is to prepare the sample. It's important to ensure that the particle being tracked is small compared to its environment so as to not interfere with its motility. And although fluorescence is not a requirement, its ability to reject the background has made fluorescent dyes, fluorescent proteins, gold nanoparticles, and quantum dots a popular option to tag the particles of interest. The sample is then imaged on a microscope, and a time series of the tracking events are captured. The images of these particles, also known as the point spread function in each time frame, is then localized. For example, in case of a 2D microscope, the particle image is an array disk, and a 2D Gaussian function is typically used as its approximate fit. This allows for the center of the particle to be localized with much higher precision than the optical diffraction limit. So in the example shown in the middle, all these Gaussian images or array disk images can be localized with much higher precision to find their centers. This also makes it so that single particle tracking is generally not limited by diffraction. The localized centers of the particles are then linked through each time frame using different algorithms based on some a priori knowledge of the expected particle movement. Dynamic information like mean square displacement and diffusion coefficient is then extracted based on either individual or aggregate trajectories. Shown here is a particle that is moving in depth as a function of time, where the color represents the time axis. Using a high numerical aperture objective as shown, the depth of field is limited to just 500 nanometer in a conventional 2D microscope. And as the particle moves in and out of focus, the localization algorithm tends to lose track of the particles, leading to broken trajectories. Furthermore, a conventional microscope is still limited by diffraction in the axial dimension, leading to loss of valuable depth information. And all this in turn leads to significant errors in analyzing the tracking data. On the other hand, our light engineering technique fundamentally changes the image of a particle such that its shape changes rapidly as the particle moves in depth. This allows for efficient capture of 3D position information of the particles without the need for stage movement. Furthermore, our PSFs are optimized to extend the depth of field to allow for particles to be in focus through a much larger volume and for longer durations. Now, all the common techniques to overcome the limitation of conventional microscopes include axial scanning and confocal microscopy. They are limited in their achievable temporal resolution and are only practical for slowly moving particles. So how do we do this? So we do this by means of a specially designed optical phase mask that is added to the backfocal plane or the pupil plane of the microscope. The phase mask modifies the PSF of your microscope to a desired shape. So in the example shown here, imagine that your particle was moving from green plane through the pink plane to the blue plane, right? So the single particle moving through these three planes where the pink plane is in focus and the green and blue plane are out of focus, or imagine three particles being tracked simultaneously. With a conventional microscope, the images of these particles look like as shown below. So the particle in pink is in focus, whereas the green and blue particles are out of focus. So essentially, the same amount of light intensity is now being spread onto more pixels as the particle moves out of focus. This results in reduction in your signal to noise ratio. And furthermore, since the size of the green and blue blurs are typically the same, this allows for, essentially, you cannot tell if the particle moved towards the focal plane or away from the focal plane, right? So when we add our phase mask to the backfocal plane of the objective, this is what it does to the resulting images. So each of those points now look like two lobes that have a distance between them, and the orientation angle is related to the z position of the particle. So essentially, you can track the particle through all those three planes without ambiguity. And also, because the rotation is unidirectional, you can tell if the particle is above focus or below focus, and the signal to noise ratio also remains constant throughout that focal volume. Another way to look at the same example is shown here. So here we have a particle that is not moving laterally and is only moving in and out of focus, right? So it's moving towards the objective and then back. On the left is a standard microscope image. In the center is a double helix image. And on the right is another PSF that we offered, the tetrapod. And what I want you to notice here is how long in focus this double helix and tetrapod remain compared to the standard microscope, which blurs out very quickly. But of course, no particle tracking experiment is just in Z. So here is another example of a particle moving through about 3 microns of depth of focus. On the top, we have its image using a standard microscope, and at the bottom, the same particle's image using a double helix microscope. And as you can see, as the particle moves through 3 dimensions, the standard microscope loses information very quickly, whereas the double helix remains in focus much longer. Also, because the double helix is continuously rotating through focus, it gives you axial super resolution because you can extract the 3D position with much higher precision. So you now have axial super resolution as well, along with lateral super resolution. Because the particle remains in focus much longer, we give you extended depth information. And all this, again, is done without any need for scanning. The rotating PSF is one example of what we offer. We do have a library of phase masks that allows users to match it to their application. And each mask is optimized for precision and depth range, with depth range extensions from about 4x to 30x that of conventional imaging systems. We even offer multicolor phase masks that show 2 different PSFs based on the emission wavelength. This allows you to simultaneously image 2 different features of your sample or track 2 different labeled particles through 3D. In order to allow access to our library of phase masks, we have developed a spindle family of products, specifically the spindle and the spindle squared. They're both upgrades to existing microscope systems that enable 3D imaging, including 3D single particle tracking, 3D single molecule localization microscopy, and 3D whole cell imaging. They can easily be attached to most scientific microscopes and cameras using C mounts, with most of our users reporting installation under an hour. Our phase masks can be easily swapped in and out of the spindle and the spindle squared to match your application and microscope setup. They offer additional filter slots to further filter out the emission light. And by removing the phase mask, you can return your microscope to its normal 2D operation, essentially the bypass mode. Diving deeper into how the spindle works, essentially, once you know you're doing a single particle tracking experiment, you would call us and we help you identify the phase mask that best matches your needs. Once we identify that, the phase mask is inserted in its holder, and we pre-align it and ship it along with the spindle system. And then it takes you about an hour or so to install this on your microscope. The spindle square is our latest offering that allows simultaneous multi-channel imaging on a single camera. So in the image shown here, the light coming from the microscope is shown as a yellow beam, which is split into two channels, the red and green channel. And the splitting can be done either using a dichroic beam splitter or a regular beam splitter based on intensity, or a polarizing beam splitter using polarization. Each of those channels can then further be modified by our engineered PSFs or left alone if that's what's desired. And the light can further be filtered using emission filters or just a linear polarizer in each of those channels. The channels are then combined back onto a single camera, making use of the large chip sizes available in today's cameras. To give you an example of what this would look like on your microscope, here is what the spindle square looks like compared to all the other components. So as you can see, it's on the order of the size of the camera, saving you valuable space on your optical bench. The data obtained using our phase masks and the spindle module can then be processed using the 3D Track software. The software has been developed as an ImageJ plugin to shorten the learning curve for most of our life sciences customers. So you would capture the data using the same tools that you're familiar with on your microscope and then import them to 3D Tracks for processing. So to sum up the features of the spindle and the spindle square module along with our software and phase masks, they're both compatible with most wide field microscopes, objectives, cameras, and emission wavelengths. They're easy to install, allow easy switching between the masks, and they're passive devices with no electronic parts. The small footprint saves valuable bench space, and they are multi-channel, enabling a broad range of biological experiments. They use a single camera, reducing cost and complexity, and they're multimodal in the sense that they can use multiple phase masks, polarizers, bypass mode. Now my colleague Scott Gomer will show you a short demo of our 3D Track software and the 3D tracking module. The data shown here was collected using a spindle module and the double helix phase mask. We appreciate Dan Schwartz's lab at CU Boulder for giving us the data to show in the demo. And in the next slide, I'll give you a quick overview of what you should expect to see. So the results from this data was published in a 2019 paper by the Schwartz group. And in this tracking example, the research group was studying transport processes through nanochannels. This is a critical step in particle separation with applications ranging from water purification to RNA movement through nuclear pore complexes. And to do this, the researchers designed an interconnected 3D structure of hollow glass spheres, also known as an inverse opal structure. The figure on the far left shows an SEM image of the hollow structure. And by tracking particles in this structure, they're able to observe how particles interact with the surfaces and escape to the next cavity. The figure on the right shows a 3D scatter plot of the particle movement with time encoded as color. And as it can be clearly seen, that a particle tends to traverse the entire sphere before escaping to the next one. Doing this in 2D would have resulted in projections of the same data, missing that information about how the particle travels, and also leading to errors in calculating the diffusion coefficients and dwell times of these particles. And with that, take it away, Scott.
Thank you, Anurag. I'm going to share my screen. Okay. So as Anurag said, we're going to go through the steps of analyzing and tracking the particle tracking experiment that Anurag just described. So here I have the wide field data that was collected using the double helix PSF of particles traversing the inverse opal structure. And I just first wanted to scroll through the data so you can get an idea of what the data looks like. So here I'm just scrolling through the movie, and you can see that there's this primarily one particle that was imaged throughout the movie. And this is a wide field image, and even though this is an engineered point spread function, a double helix PSF, it's still diffraction limited. So we can't see anything too interesting about the trajectory or course of the particle, but we do see that it's moving around, and it appears to be undergoing diffusive or Brownian motion. Okay, So let's go ahead and start analyzing this data. So here I've input the imaging system parameters for this experiment. And here I have a calibration image, which we take on the microscope in order to give the software a way to characterize the defocus information of the point spread function. So I'm going to draw an ROI around one of these bead images, like that. And I'm going to refresh in the software so that we choose the calibration image. And I'm going to input the Z range. So in this case, the depth of the calibration data and the rotation of the double helix point spread function is 4 microns. So I'm going to put in, sorry, +2 to -2. And I'm going to hit run. So the software will go ahead and fit the double helix point spread function through Z, and it will display the results of the fitting, and then it's going to calculate the Kramer-Rao lower bound baseline, which is used later in analysis to assign uncertainty metrics to the localizations. So here we can see that the PSF was fitted throughout Z, which gives us our defocus information. I'm going to close that. Okay, so we can go ahead and save the calibration, and I'm just going to overwrite my previous file. Okay. So now I'm just close this, we don't need this anymore. So now focusing back on our movie, what we want to do is we want to do the localization analysis on this. So I'm going to choose the sparse emitter algorithm. And again, I'm going to refresh and make sure I've got my movie image selected. And I'm in 0.4 for the detection threshold, making that a bit more aggressive. So in this case, it's quite sparse, and I want to be aggressive about locating as many of these PSF emissions as possible. And since it's a short movie, it won't take too long. So I'm going to go ahead and hit run. Oh, sorry. I'm going to hit run. Okay, so this starts. And this is going to take a minute to run. It's not too long, but in the interest of time, I'm going to go ahead and stop this. And I'm going to switch over to a previously saved file. So here from the double helix menu in Fiji, I'm going to choose 3D Tracks Analysis. And here we have our analysis plugin. And I'm going to choose the tracking tab and open our previously saved result. Okay. Which didn't open. Okay, there it is. Now, what we see here is we have this table of data, and we have our 3,600 frame movie. And what we want to do is we want to track it. So what one thing we know about the inverse opal structure is that the void spaces are on the order of 250 or 300 nanometers, and they are interconnected by nanopores. So I'm going to put in 500 nanometers for the lateral extent, so as to capture when the particle jumps to an adjacent nanopore, and I'm going to put in 2 for the frame gap because I expect that we've localized this pretty consistently throughout the movie because it's a strong signature. So I'm going to go ahead and hit track. Now we see these 2 columns in our table have updated the track numbers and the track lengths, and we see that there's one particle that was tracked through 3,600 frames, which is the entire movie. Particle number, or track number 2 was assigned to that particle, and it happens to be this long one. I'm going to go ahead and clear the overlay from the analysis step. Okay, so now making sure we have the right image selected here for our track overlay, I'm going to go ahead and hit overlay. And we now see the tracks are overlaid on the wide field data. And there were a couple of other beads that are generally farther away out of the field of view, but they did come in view a little bit. And then we have this one long-lived track, which is rendered in blue. And again, from this view, you don't see very much interesting about this track. It just looks like a 2D track, and it's all fairly tight together. So I'm going to zoom in on that to see if we can see a little bit more detail. So as I zoom in, suddenly you start to see something interesting. There's actually a pattern in the track, and it looks like there's kind of these spherical patterns in here. So that's pretty interesting. So let's try to get a little bit more information about that. So I'm going to switch to rendering the track color by Z position rather than track ID. So now I hit overlay, and all of a sudden I see that there's different colors which represent different Z positions or depth. So the orange is different from the yellow, is different from the green, is different from the blue. Visualize that there is indeed these kind of spherical shapes in the data, and I can see that some of the different colors overlap the others. So something's going on where the particle traversed void spaces which were at different Z positions atop the others. So I'm going to go ahead and change this now to backward and I'm going to put in 3600, which is the whole length of the movie, meaning I want to see everything that happened in the past to the beginning of the movie from wherever I'm looking. And I'm going to hit overlay. So at the beginning of the movie, which is where I am now, all I see is the beginning of the track. And as I scroll forward, the track builds, and you can watch as the particle traverses the inverse opal. And you can see that it generally tends to bounce around many times in a particular void space before it escapes through a nanopore into the next void space or an adjacent one. So as we get to the end of the movie, it starts to come back in void spaces that are on top of the previous ones in Z, so they're closer. So you can also look at it as it goes backward in the other, or goes in the other direction, if it helps you visualize. Okay, so that's what you can do with the track visualization there. One other thing I can do is I can now save this data as a new CSV file. I give it a new name so I don't overwrite the previous one. That's that. And then I can see that I now have that file, which I can open in Excel or any other software that's capable of reading a comma-separated values file, a CSV file. And again, I can see here that all of that track information is available, and you can take that track information and analyze it in whatever tracking analysis package you like. Okay, thank you, Anurag. Back to you.
Great, thank you, Scott. Let me share my screen again. Okay, hopefully you guys can see that and hear me. Thank you for the great demo. The inverse opal was one example of where the researchers were able to use our technology to discover new science. There have been a lot of different papers on life sciences, material sciences, some fundamental sciences, and all of these are available on our website. And in the next few slides, I will show you a few selected examples from these. So yeah, in the first example, this is another paper by the same group where the authors were trying to address fundamental questions regarding interfacial interactions between solid and liquid interfaces. So how does a molecule interact at those interfaces, essentially? Theoretical predictions have suggested that molecular motion at these interfaces may be dominated by hypothetical hops, but a diffusing molecule reabsorbs after giving a hop. So at the bottom here is an artistic rendering of what that looks like. This was created by the authors of, the editors of the journal who picked up this paper for a Viewpoint article. So essentially what they're showing here is that a molecule tends to take this hop and then comes back to the surface. And this is kind of a very interesting property that was predicted, but was seen for the first time in this paper. Okay, so essentially these findings explicitly demonstrate that interfacial diffusion is dominated by biased 3D Brownian motion. In the next example, this is a life sciences example by the Lee group at Cambridge. And in this paper, they combined single molecule localization microscopy and single particle tracking with the double helix PSF for studying eukaryotic cells. Unlike prokaryotic cells, eukaryotic cells are more complex and much larger. So the cells they were studying were about 10 to 14 microns in size. So that's why they decided to use the double helix PSF due to its extended depth range. So on the leftmost, you see an embryonic stem cells where they tagged the nuclear protein CHD4 and tracked it within the nucleus of the cell. And in the middle is our T cells, where they tracked either membrane protein TCR or cytoplasmic protein ZAP70 and tracked it on the surface of the cells. In each of these cases, the depth range that was tracked was about 4 microns or so. And in the case of the T cells, the focal plane was 10 microns deep into the T cell. And it's clear that the nucleus, the movement of the protein inside the nucleus is going to be 3D, and also the surface of the T cell is spherical, so all that motion is going to be 3D as well. They even did an experiment where they took all the 3D positions of the particle and basically removed the axial information and then plotted what the 2D versus the 3D mean square displacement curves would look like. And that's shown on the right. And clearly there is a loss of information and errors in the results if you were to do this in 2D. This is another recent paper by the Sheckman lab at Technion in Israel, where they combined imaging flow cytometry with multicolor 3D single particle tracking to identify heterogeneity in live cell populations on a much faster timescale than was possible before. And as I mentioned earlier, flow cells allow one to study individual cells on a much faster timescale with decreased costs and increased reliability. So in this example, they were studying drug penetration inside T cells. And to do that, they combined 3 different techniques here. So essentially, T cell is spherical in nature. And what they did was they used one fluorophore emission color to tag the surface of the T cell, used bright field imaging to find the XY center of the T cell, used the Z position of the emitters on the surface to find the Z center of the T cell. And the drug targeting particle carrier here was liposome nanoparticles, and they tagged that with another fluorophore. And inside a flow cytometer, they were studying where on the surface does these nanoparticles attach or how deep do they penetrate. And now because they have a priori knowledge of the center of the T cell and the T cell being spherical, they could do co-localization studies for drug targeting studies in this example. In the next example, the authors were looking at how different proteins signal a cell's fate. In this case, the cell was Calyobacter crescentis, and they were studying cell division. The cell forms a gradient of the protein POP-Z at the pole end of the bacteria, and then they show the migration of the various proteins away from the pole. So essentially, you can see this is the cell division process. And at the pole end of the bacteria, there is a protein called POP-Z, the bright super resolved part. And what they were studying is kind of migration of these proteins away from the pole end during the cell division process. So again, here they combined single molecule localization with single particle tracking with an extended depth using the double helix PSF. In my next example, I will show you how our multicolor phase masks can allow you for simultaneous single particle tracking in 3D. So the image shown here is from a standard microscope. And essentially, there are 2 different fluorophore colors here, green and red. But using a standard microscope, there is absolutely no way to distinguish between those 2 colors. And also, you cannot find their 3D position either. When we insert a multicolor phase mask, though, all the green particles have a vertical orientation, and the red particles have a horizontal orientation. And these 2 orientations will rotate as the particle moves in 3D. So by essentially using a single phase mask in our spindle module, you can simultaneously do multicolor imaging in 3D. And then when tracked using a 3D track software, this is what the movie looks like. So essentially both those particles are being localized at the bottom left, and animation is being created on the right, where you can see how you can co-localize the particles and study the different characteristics of the two different fluorophores. In my last example, this was a paper from Dr. Murner's group at Stanford, where again, they combined single molecule localization microscopy with particle tracking. But here, particle tracking was used to track a fiducial. And the single molecule data was collected using the double helix PSF in about 2 to 3 micron slices. The stage was then moved, or they also combined this with light sheet microscopy. So the light sheet was illuminating about a 3 micron slice near the cover slip. Collecting the data of single molecule using the double helix PSF and tracking a fiducial using the tetrapod PSF, then moving the stage to illuminate another 3 microns of the cell, and then combining all those localizations based on the location of the fiducial marker. They were able to generate these beautiful images. And so on the left, you can see that is the entire nuclear lamina of a HeLa cell. And on the bottom right, you can see the mitochondria of the same cell. So by doing this, not only do you get an entire whole cell image at the same time, but you can essentially zoom in and study the finer details of the cellular structure by combining these techniques. Just want to remind everyone that we do offer both these phase masks. We also offer the multicolor phase masks and the spindle square to potentially perform these kind of experiments. With that, I would like to thank all of you for your time, and we're happy to take your questions now.
Thank you so much, Anurag and Scott. Really